All right, here we are at week 3 for Genetics Lab 3311. My name is Ricky Johnson, and I'm your TA. So let's get started with week 3 PowerPoints. So we are doing WETM, half scoring, virtual. Don't worry too much about that. Just you'll need to learn to score. That's part of today's assignment. Don't forget that WA1 has been assigned. I will also be producing a chi-square video, as well as a video having to do with being concise, how to write the WA1. That video is done and already up on YouTube. I'll send out an email later with all of this week's videos on YouTube. We also will introduce balancers a little bit this week. That might come later as a fourth video. That one is not absolute yet, but it's not needed for the writing assignment. But you do have to learn about it for the exam. All right. We aren't going to do the double balancer, but it will be on the test, so you need to understand what double balancers are. That's all we're saying here. You need to understand how they work. Here is a link. Check out these PowerPoints. Check out that link. I will be doing an additional video on double balancers later in the, if not this week, next week. You don't need to know them quite yet, but eventually on a test you'll have it. You need to understand about maintaining healthy cultures and how to select female virgin flies. All right, so fly husbandry. All right, make only what you need. The idea is your flies can grow out of control. You don't need a billion flies always to do your experiments. Sometimes you only need a few, and you just maintain those few. Always good to have a few backups, but you don't have to keep 50 million flies flying around your lab. You need to also keep in mind that how to do crosses. I have videos on crosses that are on YouTube, and I have all types of crosses there, which will help in this course to some extent. Probably help a lot more if you're taking regular genetics. These are just things that you should have learned in biology, intro to biology, but if you didn't, you can go back and review those things like codominance and things like that. All right, you need to review this sheet because this is kind of a mating scheme. It tells you how long you're going to have to keep the flies in the vials together in order to get the results you want. So make sure you review this. This is essentially a mating scheme for what we call WTM1. All right, wild type cross 1. So go through this. Here we go is WTM2. So if you see these letters in that number, WTM2, this is what it's referring to. And these are the laboratory procedures. This is how long you would have to keep these flies together in a vial, do this, whether or not you have male or female flies for the mutant or the wild type. If you'll notice in this one, what do you have? You have, you have the mutant, and this is female. This little arrow here, that means male. So we have a wild type male crossed with a mutant female. All right, now notice the number, 3 to 10 flies of each type. You put them in a vial together, and then you wait a few days. All right, you watch them, you do you do various things, you're watching them, you're checking to make sure everything is going. You should see pupil development here, and then you should see these things. If you don't see these things, you've got something wrong going on. All right, and then of course when you get your results and you see how many flies you have of a particular type, then you will actually do a chi-square analysis. All right, if you do not have at least one WETM1 and one WETM2 cross completed this in week 3, contact your TA. Now what we do is we do these virtually online in this course. We don't actually grow them. All right. The slides here just to help you explain to you how you would do this virtually, or I should say not virtually. We're doing it virtually. These slides are to explain, if you were in the laboratory, how you would be maintaining these flies. So we're still kind of learning about it. We're just not doing it. All right, continue. These are just the instructions, what to do. It's really, we're just going through what we say do here in these various places. It's just being more explicit here, writing it out into a single slide. All right, now how do you score? Well, scoring is, well, you look at the flies and you say, well, how many males and females do I have? You have to learn how to do that. You have to look at the fly and learn to figure out what's a female and what's a male. You have to be able to identify your mutant. Is it got a funny shaped wing? Does it have the wrong color eyes? Is it the wrong color body? Does it have, extra parts somewhere? Maybe it has 4 eyes or something like that. Then you have to figure out if these phenotypes, if the allele is a dominant or recessive, and then figure out if it's a sex-linked or autosomal. And you only do that, the last two there, the dominant, recessive, and the sex-linked, you only figure that out by crossing them with wild-type flies. So you're going to have to take your mutants and you're going to have to cross them with some wild types, and then you figure out whether or not you have dominant or recessive and or sex-linked. Now you can be both. You can be dominant sex-linked, recessive sex-linked, or you can be autosomal recessive or autosomal dominant. So these two do interact. So you have to actually cross them, and you see what numbers you get after you cross them. In other words, what do the children and the grandchildren look like, and the F1 and the F2. You look to see what those results are, how many of each type of fly you have, and that will tell you if you have a dominant or recessive trait or if it's sex-linked or autosomal. Now this is actually how you do that. Do any of your progeny have the mutant phenotype, yes or no? Then if no, then your mutant must be autosomal recessive. All right. Does your phenotype have, if it's yes, and then you ask, are there females? No, then it's sex-linked recessive. If yes, it's dominant. All right, so this is kind of your flow chart for figuring out exactly what I was talking about. You have to cross your flies and then figure out after a couple of generations what it is you have. All right, now here in writing assignment 1, we're going to describe your phenotype. That means the characteristics of your mutant. What does it look like is essentially what we're asking. And you compare them to the wild type. Now, of course, you're not growing them, so you can't observe them. You're going to have to look it up online, and once we give you the mutant, you're going to have to go figure out what all these traits are. And that includes life cycle. The life cycle, some of them, it takes them longer to pupate, for instance, or they don't like to mate, so mating is slow. Some of the mutants, they don't fly very well, and actually they prefer to be up on the sides of the wall, I guess, when they're mating. And if they don't fly very well, they don't go fly up there. There are lots of reasons why they might reproduce slowly. You would normally need to observe that and talk about them. Now it says give them a nickname. We don't have to use a nickname this semester. You kind of know what you've got. Identify 4 possible genes that might give rise to your phenotype. So you get a general phenotype, kind of like white eyes. All right, but there might be more than one way to get white eyes and multiple mutations, and those different mutations might have led to white eyes plus short wings, or white eyes and they're smaller in size, versus another one which is white eyes and they're still the same size as the wild type. Well, you have to observe all of these things, and with the combination of traits, so it's more than just one, it's more than just white eyes. You have to go and find out exactly the combination of traits or characteristics, if you want, that your fly has. And then you go to Flybase right here. All right, right here's Flybase. You go there and you figure out exactly what your mutation is. All right, that's what you have to do. And that's not an easy thing to do. All right, you'll need to provide a good figure and title with your WA1. So in other words, once you figure out what you've got, you get a picture of it. They'll be on Flybase. There are example pictures. and you'll download it and use it in your paper. All right. Formatting, 1 to 1.5 pages max. All right, that's of the text. 1-inch margins, 12-point serif font, Times Roman, or Calibri or Helvetica. Arial is a large font, so if used, you have to go back down to 11 point. So these are your requirements. Has to have one figure showing wild type, has the proper figure legend, all right, and you've seen examples of that in the PowerPoints and in me giving you examples. 5 references max for this one. You might, though, collect several for later on. You might not use them all here. I mean, if you reference something, you do have to put in the reference. So this is going to limit your size. But if you find multiple ones, you might want to keep those on the side and say, hey, I'm going to use that later. That's going to be something I can say in the paper, and here's a good reference for it. Just remember, for this short one, no more than 4 pages. All right, and it's due this Friday, or Friday the 12th. Sorry, that's about 10 days from now. All right, so catalogs. This is Flybase. I went over this in a previous demo for the previous PowerPoint. You can go over that. I'll probably do at least one more of these on Flybase. Chi-squared analysis, I'm going to do a whole video on that. That will be coming in when I send the email out. For this one, I will have one on concise writing, and I will have one on chi-square. So there will be 3 videos, and one of them will be this type of analysis. And by the time you watch my video, you will understand what this means. Actually, there is a video already out from last semester that you could watch that's on chi-squared. This is the formula, all right, and this here means chi-squared. It's not quite an x, it's a special Greek symbol, chi. So chi-squared is equal to the summation, all right. Now the summation means if you have multiple O's and E's, you add them together. So there might be 5 values, 5 x's, x1, x2, x3. So you did this 5 times. You did the experiment 5 times. So you have 5 O's and 5 E's. Well, you take x1 that for O and x1 for E and subtract them, square them, and divide by the x1 with E. And I'll describe this, but the summation just means do this. You might think of it this way: do this 5 times, or do what's in here do what's in the equation 5 times if there are 5 data points. And I will do this as an example. The expected is what you expect by theory, and then observed is what you actually result. So the O is what you actually saw. The E is what the theory, your model, says you should be getting. All right, sigma means a summation. All right, and O is our experimental results. E is the expected count from the null hypothesis. Notes that you must use the number of individuals and not the proportions, ratios, or frequencies. That's correct. This is a goodness of fit test, and it works well. The chi-square, it tells you whether or not one group of numbers can be considered in the same, shall we say, group, the same population. That's the correct word, is population. Can one group of numbers, are they so far off that you don't think that they're like the others? They're in the same population as the others. So let's say you have a whole bunch of cows, but you have a few cows that are a little weird, and you're allowed a few weird cows in your population, but I have like half of my sample, my observed frequency for my sample, half of them are weird. Are they is that too many weird ones for me to consider this the same population as the larger group? And if it is, if there are some statistics, chi-square is one of them, that can tell you that, and they can say, no, this group does not belong with the bigger population. There are too many of these mutants in them. All right, so I'm using weird, but mutants are example. And so you would say they're too far from the expected model. You should only get 1 out of 10 cows that look this way. All right, and you're getting half the cows look this way. These are not from the same population. That's kind of what chi-squared can tell you. It only compares two groups, usually a global population or a large population, and then like a sample. And that's really all you can do with chi-square. It's not meant to do much more than that. And that's what it's saying. Differences between sample proportions are due to chance or from random sampling. All right. And if it's too off, if it's statistically significant, the differences, I mean, you're allowed 1 in 10. That's okay. But if you get half, 5 out of 10, no, that's way off. These two groups are not the same. That's all chi-square is telling you. It's telling you whether or not two groups have the same proportions. Okay, and if the expected one, the one that your model says you should only get one, say, brown cow out of 10, roan cow. Roan is kind of a red color. I'll say one white cow out of 10 roan and 9 roan cows. 1 out of 10 should be white. Well, then if you get half of them white, nope, that's a different population. That's not the same one. Those proportions, 1 out of 10 versus 5 out of 10, that's too far off. We can't consider them the same. All right, and let's see. I think that's it. Oh, here we go. We've got more. This is just an example. If you're expecting this and you get this, if you have this is 3 types. So you have a plant, you have 3 colors of plants, and you're expecting equal numbers of each color. But in this one, this B color is observed more often. Is that so far off that we would say that this group is not the same as this group? Maybe. We just have to do the math and find out. All right, and this is how you do the math. We get observed, all right, and here's your expected. So we do 1 trial, 2 trials. We did 3 trials here of 400 examples, and so we expect to get a proportion of 3 to 1. So 300 to 100, so the proportion is 3 to 1. Here it's 29 to 11, or 2.9 to 1.1. You can think of it that way. 300 to 100 or 290 to 110, same idea. It's a proportion. And that's pretty close. Here it's exact. But this one is quite a bit away. We know this one is in the same population. This one looks close enough. It's probably in the same population, but you never know. You have to do the math. This one looks very suspicious. I'll bet if we do the math on this, which is the chi-squared, that this one is, they're going to tell you, nope, we have to throw this one out. All right. All right. Now, there is a law in statistics called the law of large numbers. And all that law means is that the more observations you have, the more accurate your answer. And in this case, if we don't get what we want, but we only took a sample of, say, 10 flies, that might not be big enough. And you can use that to figure out how big of a sample you need. But maybe we have to measure it and do the experiment many, many times. And if you do have to do that, like we should on the previous one, we did 3 trials. Maybe it means that we have to do this 10 times. We have to do 10 trials or 100 trials to get it accurate enough so that we can be sure that we are correct about the nature of our flies. All right, no more than 20% of the expected counts are less than 5. All right, so in other words, you need at least 5 flies. All right, all individuals are greater than 1. You can't have a count of 1. I got 1 fly. No, you got to have at least 2. Okay, and there need to be 5 total, and that's of each expected count. And expected counts should be in a 2x2 or greater table. That's just when you do this table needs to be at least 2x2, which means here. But if you fail any of these, you have to use a different test. Alright, is this it? I think this is it for our PowerPoints. Oh, and expect a whole thing on chi-square. Oh no, there's more. I don't know why I wasn't showing that up. I will do, this one's getting a little long. I'm going to do another analysis finishing up these PowerPoints.